Hello everyone, my name is Prothama Manna and I'm going to talk about my master's project work on free-ranging dogs vocalization. Animals use a wide range of sounds to communicate between themselves. These sounds are often used for territorial maintenance, to attract mates, or as an alarm or warning call. Dogs have a broad range of vocal repertoire. Bark is the most common type of vocalization used by them. Barks are the short and repetitive signals which also differs between breeds and even between individuals. Several studies have been carried out to understand communication in dogs, but little is understood of this behavior in a natural environment. Vocalization is mostly used for social interactions. Thus, it is best studied in group living animals such as free-ranging dogs. These street-living dogs stay in a group and they largely depend on humans for food. These dogs are highly territorial and actively use vocalization to maintain their social dynamics. Free-ranging dogs not only exchange barks among their group members and the neighboring groups, but also with the groups that are out of their visual range. However, it is not understood in natural environment and in a context-independent situation whether the free-ranging dogs can differentiate between their own groups barking and other groups barking. We tested 40 groups with a minimum group size of 3 adults in 4 different urban locations. The experiment was done in 2 phases, the recording phase and the playback phase. On the recording phase, we went to the territory of each group and recorded all the vocalization of the group that occurred during a period of 1 hour using a voice recorder. The barking track, which had the least background noise and was for the longest duration, was chosen for each group. In the playback experiment, 3 tracks were played for each group, among which one was their self-group barking and the other 2 were from 2 randomly chosen different groups. Total 3 trials were done on each group and the tracks were randomized in every trial with a 2-minute settling time between the tracks. The tracks were played using a Bluetooth speaker kept at a 3-meter distance from the center of the dog group. The speaker was hidden under some bush so that the dogs can't see it. All the responses were recorded using a video camera. The result reveals dog groups did not show significantly different levels of response to self and other groups barking. The latency to respond and the response duration were comparable in self and other conditions. The latency shows the urgency of the dogs to respond and the response duration reveals the intensity of the response. Responses were categorized into 3 major behavioral types: change of state, movement and vocalization. Vocalization responses were further categorized into vocalization 1 and 2 according to the experimenter's perception of energy expenditure and aggression. Vocalization 1 includes less aggressive vocalization like barking, howling, whereas vocalization 2 includes more aggressive vocalization like angry barking, barking back, etc. The nature of the response in self and other conditions was comparable and the change of state was the most common type of response. The overall responses of males and females were also comparable. To check if the tracks were comparable in their acoustic signatures, 10 randomly chosen syllables or single barks from each track were analyzed for 6 different acoustic parameters. PCA was carried out using all 10 syllables of every group. Here each color represents each group and each dot between these are the syllables of the bark. Further PCA was made considering locality as a variable. Both the PCA plots revealed clustering of the tracks irrespective of group and group's area identity. To sum up, the free-ranging dogs showed similar response to recorded barks of their own and other groups. There was no gender bias in the response. This directly challenges the common belief that the alpha member of the dog groups is always a male, who is the most aggressive and the reactive member of the group. The response when observed mostly consisted of alertness rather than more energy-intensive responses like movement and vocalization. Thus, it is likely that the dogs were not much perturbed by the playback sound of the bark that was presented to them out of context. Our spectrogram analysis revealed that their barks were largely overlapping with each other in the PCA space. Thus, it is impossible to distinguish between the different barks analytically. This result, when coupled with the responses of the dog in the playback experiment, is interesting as it points towards an essential role of context for their behavior. It thus seems reasonable that the dogs were not able to distinguish between the different barks when no context or additional cue was provided to them. Perhaps a combination of visual, olfactory and auditory cue is required to produce a stronger territorial response to the barking. Thanks again for watching and I will be happy to answer any question during the Q&A session.